Hi everybody. So today we're going to take a look at two different case studies. Our first case study is a man named J. Wellington Wimpy. Wimpy has struggled with his weight his whole life and has just kept gaining weight due to his diet. So his previous diet contained mostly burgers, but after an intervention with his friends about his health, that all changed. His new diet now consists of turkey and veggie subs, and he has even started exercising with some of his friends. Before Wimpy's transformation, his diet consisted only of burgers. These burgers contained mainly simple carbohydrates like bread and cheese, and very little complex carbohydrates like vegetables. This diet did contain some protein from the burger, but this protein contained lots of fat. The simple carbohydrates in these burgers are used for instant energy, so they're broken up by the body very quickly. Because these carbohydrates are broken up so quickly, their energy is spent very fast. Because of this, he will be hungry much quicker than he would be if he had a mainly complex carbohydrates diet and will need to eat more food to feel full. After his change in diet, his turkey subs became more oriented towards complex carbohydrates. They still contained some simple carbohydrates such as the bread and cheese on the sandwiches, but they had lots more veggies on them and therefore were much better for Wimpy's diet. They did contain proteins in the turkey, but this protein contained much less fat than the burgers did. This diet that contains more complex carbohydrates will allow him to feel full longer and have more energy throughout the day. Because of this, he can actually eat less than he would with the burgers because they will give him the same amount of energy from the better carbohydrates. These complex carbohydrates take longer to be broken up by the body, but their energy does last much longer than the simple carbohydrates would. On this slide, you can see a maltose molecule being broken down. So maltose is a disaccharide that breaks down to a hydrolysis reaction. For this to happen, the molecules must be in an environment with water to break down the glycosidic bonds between the two monosaccharides. Once this bond has been broken, the body can digest this monosaccharide and use it for energy. These disaccharides will give more energy that lasts over a longer period of time, so they're better for Wimpy's diet than the monosaccharides. Disaccharides can be found in vegetables and other parts of Wimpy's new meal. Carbohydrates are used in our cell as a primary energy source, and they also serve as fiber. Carbohydrates contain a carbonyl functional group which allows for them to bond with other molecules and also allows for them to be broken down from their ring state into linear molecules. Both of Wimpy's meals contain protein, which have many different functions in the body. When protein is digested, the peptide bond is broken. For this to happen, water must be added. Once the water is added, the bond will break through a hydrolysis reaction, which is pictured on this slide. The protein will then split into its amino acids, which help Wimpy's body in many different ways. In our cells, proteins can be used for many different things, such as framework, fighting infections, sending messages, transportation, cellular markers, enzymes, and movement. In protein groups, there is a central carbon atom. Attached to this central carbon atom, we find a carboxyl group on the C terminus of the protein and an amino group on the N terminus of the protein. These groups are attached on either end of the central carbon and help to create bonds between proteins and hydrogen bonding within. Also attached to the central carbon is a hydrogen atom and an R group. This R group can be simple or complex and is responsible for creating ionic bonding, hydrogen bonding, and disulfide bridges in the tertiary structure, which help to form the protein's final 3D shape in the quaternary structure. Proteins have 4 different types of structures. The first structure is called the primary structure. This is a linear chain of amino acids in a polypeptide chain. The next structure is called the secondary structure. In this structure, the protein begins to take its shape due to hydrogen bonding. In this structure, you can have 2 results, either an alpha helix or a beta pleated sheet. The next structure is called the tertiary structure. This is when the overall shape of the protein starts to form. In this structure, bonds start to occur between the R amino acid groups. This can result in ionic bonding, hydrophobic interactions, and disulfide bridges. The final structure of a protein is called the quaternary structure. In this structure, 2 or more polypeptide chains become linked together. The same type of bonds that were formed in the tertiary structure will begin to form again between the new polypeptide chains. This will leave us with a final unique shape for a protein. Our last macronutrient is lipids. In Wimpy's previous diet, he had a lot of fat from the burger. Now in his turkey subs, he has much less fat. This fat is healthy for you, but in large amounts it can become very dangerous. On your screen now you see a triglyceride, which was most likely present in very large amounts in his old meals. This triglyceride cannot be digested as is by the body, so a hydrolysis reaction occurs, which breaks down the ester links by adding 3 water molecules. When broken, the triglyceride becomes a glycerol molecule and 3 fatty acid chains, which are stored in adipose tissue in the body. This fat will be later used as a secondary source of energy, but if it is not used, it will build up as weight gain. In the body, lipids are also used as a secondary energy source to create membranes, such as cell membranes, send chemical signals and hormones, and also help provide structure and protective features. These functions tell us that lipids are good in proper amounts and should not be cut out of our diet. Yet, when we have too much of this secondary energy source, it becomes stored as excess weight. Lipids contain a carboxyl group, which are used to help them bond with fatty acids in structures such as triglycerides. They also contain a carbohydrate chain, which is made of carbon and oxygen. On this screen here, we see a lipid molecule. Highlighted in green is the carboxyl group, and highlighted in pink is our hydrocarbon chain. If Wimpy would like to continue to improve his health, he can increase the amount of complex carbohydrates in his diet while decreasing the amount of simple carbohydrates. To decrease the amount of simple carbohydrates, you can change your bread type or have less bread. A multigrain bread will have more complex carbohydrates than a white bread will. When you have more complex carbohydrates in your diet, your energy will last longer. Wimpy can also consider increasing the amount of vegetables on his sandwich and decreasing the amount of fat on his sandwich by eliminating things like dressing and butter or decreasing their amounts. Our second case study is about a woman named Carol. After recovering from a virus, Carol began to feel sick when she consumed dairy products. She visited her doctor about this issue and discovered that she is now lactose intolerant. The virus had affected the enzyme production, and so now she is not able to digest lactose. To be lactose intolerant means that your body is unable to digest the sugars found in milk. These sugars are called lactose. The body does not have the correct enzymes to process lactose. Enzymes are a biological catalyst which speeds up the reaction of digestion in this case. Because she does not have these enzymes, she develops symptoms such as bloating, nausea, and gas. These symptoms are because when lactose is not digested earlier in the digestive tract, once it reaches the intestines, it then becomes digested by bacteria instead, which produces CO2. The lactose in Carol's body must be processed by lactase, which is the enzyme that breaks down lactose, due to its unique 3D shape. Because of this 3D shape, each enzyme has their own unique active site shape. You can see the active site on this diagram at the red arrow. This active site allows the enzymes to only process certain substrates. If the shape of the substrate does not match the shape of the active site, the enzyme is unable to process this substrate. As you can see in this diagram, the matching substrate for our enzyme is substrate number 1 due to their triangular shape. Carol's doctor recommended to her that she is still able to eat yogurt, even though she is lactose intolerant. The reason behind this is that yogurt contains the enzyme lactase, so when it is consumed, it can actually help people who are lactose intolerant by giving them lactase enzyme to digest the lactose in their body. This means that yogurt is a very low-risk food for causing symptoms of lactose intolerance and is therefore safe to consume. The current model of the enzyme is called the induced fit model. This is pictured on your screens right now. This model was developed from our original lock and key theory. This original theory stated that the enzymes were like a lock and the substrates were like a key. This model was fairly corrected after further examination. Our new theory was created. This induced fit model says that as a substrate approaches, an enzyme will slightly modify its shape to perfectly fit the substrate. Enzymes are still very specific, like a lock and key. Yet now we know that enzymes can change the shape of their active sites to make it just right for the substrate they are looking for. When the substrates are in the enzyme, this is called an enzyme-substrate complex. This is pictured in the two middle pictures of this diagram. After this enzyme-substrate complex is formed, the enzyme will bind the substrates together to create a product which is then released back into the system. The amount of product an enzyme can produce is regulated by allosteric activation and allosteric inhibition. Allosteric activation is now pictured on your screen. In allosteric activation, an allosteric activator will bind to the allosteric site. This causes the active site to allow substrates to bind to it, which increases their ability to create products and puts them in a high affinity state. The second way to regulate production is by allosteric inhibition. Allosteric inhibition causes the enzyme to go into a low affinity state. The allosteric inhibitor will bind to the allosteric site, which in turn changes the shape of the active site to stop substrates from being allowed to bind to them. This puts the enzyme in a low affinity state, which will reduce the production of any products that could be made. And that is the end of our case studies for today. Thank you so much for listening and have a great day.